Hello guys, in this video we will be going through the concept of cellular respiration. What is cellular respiration? Cellular respiration is the process of using organic compounds in an enzyme catalyzed reaction step by step to produce ATP. Now, what is ATP? So you guys might have already heard ATP is the energy currency of the cell, right? Just like money is the currency that is used in real life, and to do anything in life it requires money. Same, to do anything inside the cell or inside the body it requires ATP to contract the muscles, to move ions inside and outside of the cell, to transport hormones, proteins, and even for cell division it requires ATP. So ATP is required by the body for almost every function. Now, how is this ATP produced? ATP is produced by adding an inorganic phosphate or a phosphate from another substrate to an ADP molecule to produce ATP. Right? Now, as I told earlier, cellular respiration is the breaking down of organic compounds. So, the organic compound that is broken down in cellular respiration is glucose in the human body. Now, when we consume food, the food has a lot of energy. Food has a lot of nutrition, mostly carbohydrates, lipids and proteins. So, carbohydrates are the main source of energy that is usually used to produce ATP. And carbohydrates are broken down in the mouth and the small intestine. It produces glucose. Now, this glucose molecule is very fascinating. It has a very high amount of energy. Now, if we break down this glucose all at once, what happens is all the energy that is present inside the glucose can be transferred to heat energy. So, we can efficiently make ATP using that. So, we have to break down glucose step by step in enzyme catalyzed reactions using a lot of enzymes. In that way, we can efficiently grab all the energy and use it to produce ATP, right? So glucose is the organic compound. Now cellular respiration can occur with and without oxygen. So if cellular respiration takes place in the presence of oxygen, we call it aerobic respiration, and if there is no oxygen present, we call it anaerobic respiration. But today in this video, we will be going through the process of aerobic respiration in the presence of oxygen. Now during cellular respiration, we have three main processes. First we have glycolysis. So in glycolysis, glucose is broken down into 3 carbon molecules. Then we have pyruvate oxidation and citric acid cycle, where the 3 carbon molecule is further broken down, further undergoes oxidation to produce more energy. And finally we have electron transport chains. Now, as I told, glucose has a lot of energy, and this energy is released in the form of electrons. So, these electrons can be accepted by certain molecules. We call them electron acceptors. So, one of the best molecules that is involved in cellular respiration is NADH+, and another molecule is FADH+. So, these molecules can accept the electrons when the glucose molecules are broken down, and finally they can accept the electrons and convert themselves or reduce themselves into their high energy form such as NADH and FADH, and these can be used in the electron transport chain. So we'll talk about all these in this video. First, let's start with glycolysis. First, we have a 6 carbon molecule which is glucose. So during glycolysis, what happens is this glucose is broken down into 3 carbon molecules called pyruvate. So we produce 2 3 carbon molecules. Ironically, as I told earlier, for all the reactions ATP is used. So even for this reaction, we need 2 ATP molecules to start it. So we use 2 ATP molecules here to start the reaction. And as the outcome of this reaction, we can produce 4 ATP molecules. So this reaction, breaking down of glucose to pyruvate, will give out 4 ATP molecules. And another thing is, glucose to pyruvate is not a single reaction. It is a series of reaction. So let's say glucose is broken down into another intermediate product. That intermediate product is broken down into another product, and finally it will produce pyruvate. So in this whole series of reactions, it will produce 4 ATP molecules. And also hydrogen ions are also released. So 4 hydrogen ions are released, and also electrons are released. 4 electrons are released during this reaction. Now, as I told earlier, these electrons, they contain very high amount of energy and we can't lose them. So what we do, these electrons are accepted by these electron acceptors called NAD+. So an NAD+ can accept 2 electrons and a single hydrogen ion and convert themselves to NADH. NADH. So this NADH has all the energy that was present in the electrons. So during glycolysis, glucose to pyruvate conversion, 2 NAD is converted to 2 NADH. Why? Because we have 4 electrons. 2 electrons to produce 1 NADH and the next 2 electrons to produce the other NADH. Now, in this process we have produced 4 ATPs, right? 4 ATPs, but 2 ATP was used to start the reaction. So the net yield is only 2 ATPs, right? Because 4 minus 2 is 2. And also we produce 2 NADH. Now this pyruvate, okay, before that, this reaction occurs in the cytosol of the cell. Okay, why? Because all the enzymes that is required for the conversion of glucose to pyruvate is present in the cytosol. For that reason, this reaction takes place in the cytosol of the cell. But the stage 2 and stage 3, they take place in the mitochondria of the cell. But only when oxygen is present, this pyruvate can enter the mitochondria of the cell. So let's assume the oxygen is present and the pyruvate is entering the mitochondria. So this is the outer membrane of mitochondria, and let's tell this is the inner membrane, right? Now, if oxygen is present, pyruvate will enter into the mitochondria using certain protein channels. So pyruvate. Now this pyruvate is a 3 carbon molecules. And also remember, we have 2 pyruvate molecules per single glucose. Okay. So 2 pyruvates enter the mitochondria. So this pyruvate, what happens is, they lose a carbon dioxide molecule. So now the pyruvate has 3 carbon, it is losing a carbon dioxide molecule, it will become a 2 carbon molecule. And it also releases electrons. So these electrons are again accepted by NAD+ and converted to NADH. Right? Now we have a 2 carbon molecule and electrons accepted by NADH. Now what happens is, it combines with a molecule, a compound called coenzyme A, and produces acetyl-CoA. Now this acetyl-CoA is a 2 carbon compound. So you can see, for a single pyruvate molecule we have produced 1 carbon dioxide and 1 NADH, but for 2 pyruvate molecules we will produce 2 carbon dioxide and 2 NADH. Right? Now this acetyl-CoA, what happens is, it will enter the citric acid cycle. Now citric acid cycle contains a lot of enzymes and it is a series of reactions. So in the citric acid cycle, what happens is, this acetyl-CoA will undergo further oxidation and it will remove all the remaining energy that is present inside the acetyl-CoA. So after one cycle of citric acid cycle, acetyl-CoA can produce, give me a sec. Acetyl-CoA can produce 3 NADH. 3 NADH. And also 1 FADH. As I told earlier, FADH is also an electron acceptor, and they can also produce 2 ATP molecules. 1 ATP molecules per pyruvate. Now, this is for one acetyl-CoA, but we have 2 acetyl-CoA. Why? Because we have 2 pyruvates, right? So all these are multiplied by 2. NADH multiplied by 2. So we have 6 NADH. FADH multiplied by 2, we have 2 FADH and 2 ATP. Now, the glucose molecule has been completely broken down and it has no remaining energy. Right? As you can see, we have produced 2 ATP, 2 NADH in glycolysis. In pyruvate oxidation and citric acid cycle, we have produced 2 NADH, 2 NADH in pyruvate oxidation, 6 NADH in citric acid cycle, 2 FADH and 2 ATP. And in citric acid cycle, and also 4 carbon dioxide is also released. Now, we already have 4 ATPs, right? We already have 4 ATPs. Let's write it down. Now, using this NADH and FADH, we can produce more ATPs. So, that is what occurs in the electron transport chain. So, this is a very long process. So, I am not going into details about this process in this video. So, in summary, what happens is these NADH and FADH, they are used to produce ATP. How? So, these NADH and FADH, they will give out their electrons, right? As you can remember, they have very high energy electrons. They will give out these electrons, and these electrons can be used to produce ATP. So, on average, a single NADH can produce around 2.5 ATP, and single FADH can produce around 1.5 ATP. Okay. So, we have 10 NADH molecules in total, right? So, 10 NADH molecules means we will produce around 25 ATP. And we have 2 FADH molecules, so we will produce around 3 ATP using that. So, totally we can produce around 32 ATPs using a single glucose molecule. So, that's it all about cellular respiration. And another thing is, as you can see, we have produced 4 ATP without using NADH and FADH, and we have produced 28 ATPs using FADH and FADH2 and NADH. So, producing ATP using NADH and FADH2, we call it oxidative phosphorylation, which occurs in the electron transport chain. So, we call it oxidative phosphorylation, right? But 4 ATPs that was produced here, that was produced by substrate phosphorylation. So, during substrate phosphorylation, let me explain it. Okay. So, during substrate phosphorylation, what happens is this phosphate group comes from another substrate, right? So, as I told earlier, ATP is produced using ADP and phosphate. So, if the phosphate is coming from another substrate, as I told, glycolysis and citric acid cycle has a lot of intermediates, right, in the cycle. So, these are all substrates. So, if the phosphate is coming from there to produce the ATP, we call it substrate phosphorylation. And if the ATP is produced in the electron transport chain using NADH and FADH2, we call it oxidative phosphorylation. So, that is all about cellular respiration.